welcome to the large crowd of the audience to this seminar. We are very lucky to have Dr. Gonzalo Maso Talou here from the Auckland Bioengineering Institute and the University of Auckland. Quite far from here.
Yeah.
Really kind of the other side.
52 hours of flight. Lovely.
Okay, good. Gonzalo studied computer science or kind of software engineering in Argentina, and then thank you. And then moved to Brazil. Quite interesting for an Argentinian to do the master and the PhD, and he and then moved to Auckland, where he is a senior researcher. And to me, it's a bit difficult to say on what he is an expert, because, I mean, I don't know, but I've been trying to ask him about a lot of different things, and he always answers like, yeah, I mean, obviously, so easy. So you asked him about modeling.
I was up to text all this time.
Yeah, exactly, exactly. About computational modeling, and he knows a lot in terms of mechanics, in terms of hemodynamics, machine learning. He knows a lot from long time ago also. Brain, yes. Heart, yes. And I said, okay, that's not too bad. And obviously now trying to combine all this knowledge in an integrative way. And I think that links with the title, right? That it's kind of integrative and linking brain and the heart in interesting ways. Thank you very much.
Okay.
Thank you. Talk there?
Okay, yeah. So, thank you for the lovely introduction, and I hope I'm to the level of how you introduced me. So, yeah, a lot of pressure now. Now, thank you very much, Oscar, and it's lovely to finally be here. So we collaborate a lot with Oscar since a few years now, and I always wanted to come to Barcelona. Apart from Barcelona, it's a great place, but just to meet the group and to see what you are up to here. So I hope that during these days I can have some idea, at least, of what all these people is doing. So, as Oscar said, I went all a little bit around the place in Auckland Bioengineering, that is an institute that models, with computational models, all the different organs in the human body almost. So we have captured most of the systems, and we have a good interchangeable researcher from the different groups. So at the end of the day, you know about physiology. You don't know about one organ, but you know about everything a little bit. And that's why we create a group on the brain, but you need to have an edge to work on the brain, because everybody works on the brain, and with very well-established groups for 20 years old. So we start to work on this neurovascular, physiologically integrative approach. That is what I'm going to talk to you about. This is mainly about my group. So this is the brain, most of you know, and it's the most important organ in the body, and it's one brain talking to other brains saying that, so it's no bias there. And the current understanding always is the neural brain. So we always talk about the neurons and how the brain organizes in these nice networks that they fire at these different frequencies, and they talk to each other. But what really happens in physiology is far from being only that. You have things like the glia brain, and the glial brain is talking in terms of calcium waves that are propagating through the brain, and choose what to activate and what to deactivate in the brain. And talks a very different language. It's more diffusive language. It's less spontaneous and sporadic. And there is also these questions of, okay, can emotions be portrayed in the glia, not in the neurons? And a lot of other different questions. So, but at the same time, if you are going to say, okay, neurons and glia, but there is also heavily regulated by hormones. So hormonal brain can be another approach, and so on, so on. We can talk about the vascular brain. And the vasculature is quite interesting because it's what is going to orchestrate how the oxygen and nutrients are distributed across the brain, which start to be very important when you want to model activity, because without the correct fuel, you cannot do the correct functionality. So in this point is where we said, okay, we have 10 years of research on vasculature, So we have something that most of people do not have, that is a full body closed loop cardiovascular model. And we know pretty much how to personalize that when we have enough data. So because of that, we said, let's do this. Let's start the brain with a cardiovascular approach. And obviously, it's not only performing hemodynamics and see if you have a stroke, then this part do not have blood and that is dead. It's more about how we couple what happened at the low level on the vasculature and the neurons, what happened in the systemic level in the autonomous nervous system and the cardiovascular system, and how these two big physiology systems are interacting between each other. And it's in this realm that we live, and we have all these people that we need to bring together. So people with very different backgrounds. I have a weird background, so I'm software engineer by training, but then I did an applied math kind of master's, and then I did computational modeling, that it's a mix of computational methods and mechanical engineering and math. So that gave me the edge to be kind of the glue across different specialties. Then Soroush have a heavy mechanical background. Finn Barargus, that is one of our researchers, he has a strong background in control systems. And that's why he's very important for autoregulation in our projects. But then our students are also as diverse, because we have a software engineer, we have a physician, we have a mathematician, we have a physicist and mathematician, a bioengineer, neuroscientist, and a mechanical engineer. And that is our PhDs, but also our research assistants that provide us all the infrastructure that we need to do research. They also have quite a variety of backgrounds, and that makes it very cool to work together every day, because one day we are doing something on the physiology side, and suddenly Robin said, guys, what you are doing is wrong, so we need to realign, and then the math may not be right on Robin's side, but then Cameron and Hershey will jump and say, Robin, you need to change that in this and that way. So allow us to have a very constant check on the different specialties, and also a quite interesting discussions in the group. So how we do this integrated neurovascular view. So the group that I'm presenting you is quite young, have 3 years old only. There is no PhD finished since we start yet. Not because they are not able to finish the PhDs, obviously. It's because they is quite young. They didn't have time enough yet. So this is what we want. We have the cardiovascular system. For instance, here we have only the heart and brain axis of the cardiovascular system, and we want to heavily integrate it with the autonomous nervous system. That, for those that you don't know, it's the part that is you are not conscious of how it's operating in your body, but is adjusting the different systems to maintain homeostasis and allostasis process. And that have an important intertalk with the cardiovascular system at the level of the heart through chronotropy, inotropy, ludotropy, lusotropy, dromotropy, so etc. A lot of different effects that perform in the heart, changing its properties. You have sensors of the cardiovascular system for the nervous system, that in the aortic arch you have high pressure baroreceptors. Also in the carotid sinus you have the same. In the terminal of the vessels you have the astrocytes that with their end feet they are sensing the pressure on your vascular system. So you are sensed all around through by the nervous system, and the cardiovascular system is adapted by effectors from the nervous system, and that is changing the tone of your vessels. So basically the nervous system had the ability of adapt your system to where you need blood flow or where not. So how we can develop enough technology in order to have these very complicated integrated models from for a patient-specific research was the main challenge. So our first PhD was obviously, we throw him the very easy task and say, okay, if we give you enough MRI data, you are going to construct the models without any human intervention at all and have to be fully calibrated. And that is what Zhantao Shen is his PhD about. So he started from some MRI images. Here are some MRI images, but then he used also functional imaging to calibrate the model. and segments, extract center line from those vessels. Here there is always machine learning, obviously, unit, and all the stuff that you guys know very well. Then from center lines, you move to a graph structure, and this graph structure with GNN, graph neural networks, are labeled with anatomical labels. So then we know which part of the graph is which vessel, and with that we create this bond graph that is a very convenient way of creating lamp parameter models. Those lamp parameter models are going to be fine-tuned with functional data to reproduce patient information. Yep. So this project is quite advanced. It's almost in its end. We are writing 2 papers currently with Gonzalo, and hopefully I can send you the papers in the near future. But this gave us the infrastructure to start working more intensively in the area, because now that you have automatic models, and is in itself a compiler, and I'm saying that because I'm a software engineer, that basically you throw some images and configuration files, and you have a model coming out of that. So pretty much work as a compiler. That gives us opportunity that now any student, without doing anything at all, can have the cardiovascular system working with them for a specific patient. So because that worked quite well, then we said, okay, now we have Ali that have a great understanding on mechanics and is very good at programming as well. So he will be doing something similar for what we call the Neurofluid Model. The Neurofluid Model, it's basically the integration of this hemodynamic model, as Gonzalo was generating, but also is coupling the terminals with the CSF space. The CSF space is where the brain is floating, and basically it's a very important part of the brain to know its behavior, because we'll impress different pressures on the brain, and that will lead to cognitive impairments. And usually, for the physicians to know the pressure that you have in that compartment, they have to make a lumbar puncture, which is far from being ideal, because if nothing goes wrong, anyway, it's not that reliable neither, because the system readapts as soon as you puncture the dura mater. So we are making this project to have the internal pressure of the CSF with non-invasive methods, and that is done thanks to this nice MRI. If you see here, the video is not rolling. That's awesome. Let's see if we can make it play. Okay, let's do it this way. So hopefully you can see something there. So can you see that it's moving the lower part of the brain? So with these images, we are able to characterize microstrains on the brain. Yeah, and as most of you know, the brain is composed of non-linear materials. So this nonlinear mechanics allows you, if you see the movement, to make a good educated guess on the constitutive materials. And with that, we want to characterize the pressure that is exerted on top of that. I think I have a little bit of feedback. Okay. So this is kind of what his project is about. Try from medical imaging to obtain that, the intracranial pressure. And also, as a byproduct, you have the characterization of the mechanical state of the parenchyma of the brain, which is quite cool. And then we start having the problem that there is only so much you can see in the brain with non-invasive techniques. And that is the problem of only looking at arteries of 30 millimeters at best. So we have methods that does this nice isogenesis generation for small arteries, but we still cannot move from small arteries to capillaries. And that is where Cameron Appledorn enters into the scene. And he's created methods that are based on pure biology principles. And the idea is that if you have a tissue and you know the metabolism of that tissue and how it grows through life, you are able to regenerate the capillary bed that is associated with that. Yep. So we are obviously not claiming that we are going to reconstruct the exact capillary network. That is kind of impossible. But we want to create models of capillary networks that have a similar functional response to the one that you have, which that would be good enough when you want to integrate with a macro model. So these are the main three processes that you need to figure out to build a vascular network. And angiogenesis here is lost, but you have two kind of angiogenesis. That is when you generate a vessel. So if you have a capillary like that, at some point you have, for instance, here you may have a little bit of hypoxia, and a new sprout will start growing from here. The sprout are the things like look like hand. And it's going in this region. Another sprout may come in from the venous side, and it's going to anastomose. That is the first process. And close a vessel. Yeah. And this is going to redistribute the flow. Because the flow is redistributed, here you will have higher wall shear stress because now you have to push more blood flow to accommodate the flow. And that is triggering what is called arteriogenesis. That is that your radius is going to grow. Yeah? And when your radius grow, little by little, smooth muscle cells will also make part of this vessel, so you promote from capillary to arterial. As this process continues and continues, you reach a more complex structure, but some of the vessels, when you create new connections, are going to be detrimental for the overall flow, and you will start having a stagnation or microclots. And those are going to trigger what is called the pruning, and it's the regression of those vessels. So the endothelial cells will walk back through the walls. It's a very cool phenomenon, in fact. So they crawl back through the walls of the larger vessels and they reaccommodate it somewhere else. And that process is very important because now if you think, you have a process that is growing vessel, another one that is regressing vessel, and that is pretty much what optimizes your networks. Yep. So that is the ABC. Now we need to make it work. So that is Cameron problem for the next 3 years. And as we grow in complexity, we have more vessels, we have large computational models, everything is nice, but then we cannot run it anymore at some point. So Herschel has his project in actually trying to help us with that. First, we throw, obviously, the simple problem of real-time Navier-Stokes. Why not? But what really we want from here is methods to accelerate hemodynamics. We are not that ridiculous in thinking that he's going to solve everything, hopefully, but it's good to start and start somewhere. And he's going to start doing a Navier-Stokes 3D for the cardiovascular tree. And for that, we propose a strategy that we discretize the space and the time for the pulsatile Navier-Stokes equations. And all of this will be powered by AI. So with this deep learning, these models, we already published them, are AI surrogates, and they will be solving n different steady-state problems that will approximate the pulsatile problem. That is with a frequency decomposition of the problem. And we do a similar strategy by decomposing also the space. And then the problems that you have to solve are very local and very simple. So these networks do not need to learn the complexity of a complete network that is achieved by integration. Yep. So the idea here is to have a good AI method to give a very good educated guess of what is the hemodynamics, and then you can converge it with whatever method you want. If you are not happy with e to the -2, e to the -3 relative error that AI usually brings, you can use a finite element methods, initialize from the AI, and you converge it as much as you want. Yeah, but nonetheless, it's going to be way faster than actually doing everything by finite elements or finite volumes. Everything that we talk, as always, is for all white males. Yeah, so there is a big problem because I'm not the only ones with diseases. And this is where Robin has her area of actuation in our group. She's the one creating other populational representative models. And she's starting with the newborns because she's from pediatrics. She has a background in clinic related with newborns. And she worked a lot of time in what is the Legan Institute back in Auckland, that they mainly work with very early age patients. So she chose to create the neurovascular models that we have one for an adult, but she's creating one for a newborn term, another one for a newborn preterm. That they are different because they have these blue connections. So these are extra connections, shunts that you have very early in life. And obviously, as soon as you are born, they are collapsed and they disappear. But when you are born preterm, it's not the case. So they can be functionally active to some degree. And this is her PhD, is starting building these models and calibrating these models, which is strongly based in some of the work that Finbar is also doing. So they are creating compilers now for newborns. And they achieve a very good precision in the calibration so far, but we still are, we are recruiting more preterm babies and term babies to actually have a more rich data set and see that we can generalize for a fair, large, I will not say the word large, but for a good representative population, at least. So in this study, she's generating this for male and female, as female vasculature is not different from male at this early stage in life. But it's something that we also want to analyze more scientifically. Okay, everything that I talked till now is a vascular group. So where is the electrical activity and everything around the neural brain that you say at the beginning? Are you thinking? So here is Gerling is the first PhD going in this direction, and he is using one of the models that is from your house here in UPF. So this is Gustavo Steco's model. He's working with mean fields, and he is basically approximating the neurodynamics starting from EEG data and DTI information. So DTI, for those that, of you that don't know, gives you a very good understanding of how the white matter is connecting the different parts of the cortex with also subcortical structures. And this information is used as the connectivity of a lot of simplified units that are representing a cluster of neurons. And those cluster of neurons are split between excitatory and inhibitory neurons. Yep. They have different also types of connection based on different neurotransmitter and neuroreceptors. And as output of this model, you can have a nice hemodynamic response, and that is needed because the classic way of visualizing the brain activity is using the Bolt signal. The Bolt signal, think on hemodynamics. It's basically a surrogate of hemodynamics to try to understand how the brain is activating and how it's performing. Yep. But it's not electrical activity you are looking. It's basically oxygen levels, and it's heavily correlated with hemodynamics. So once you calibrate the system with your EEG and your connectivity given by DTI, then you have 3 parameters in your model that you need to tie up, and you do that by comparing with the fMRI, and you can simulate an fMRI at the same time. So then you minimize the discrepancy between these two guys, and you get your parameters that best reproduce the fMRI, and hopefully it's grabbing the right dynamics in your system. There is overfitting? Yes, there is a lot of overfitting. But it's not necessarily bad, as we don't have any other way of doing it. So it is what it is at this point. We are working on how to make this more physiologically accurate, or if we can recover more grain, fine grain level details of the system at this point. So this is an ongoing PhD. It also allows you to have personalized models now for electrical activity. So what comes next is to say, okay, instead of just approximating a bolt based on a simple balloon wing Kessel model, we can use our big brain, both big brains, the one that we have here and the one that we have on our heads, and let's try to couple them and solve all the system together. Let's try to solve the parameters that we have free on the hemodynamics, at the same time the parameters that we have free here, such that match this. But now the match to this will be constrained by hemodynamic, only plausible hemodynamic responses. So, and that is nice, but how you connect these two guys? And that is Sergio Dempsey, PhD. Basically, he works on how is the crosstalk between astrocytes, neurons of different types, the endothelial cells that you have in your vessels, and how is the mechanisms going from the change in hemodynamics, how affect the activity, and how activity affects hemodynamics. Yeah? He's pretty much centered on dementia because this crosstalk between these two systems starts to be dysregulated when you undergo dementia and can be a very good early stage biomarker. So he's pitching his research on that side, but it's not only constrained to that. It's and the neurovascular unit you can use in healthy patients, in patients with different conditions. And it's a general enough technique to couple with our other research. And this is where everything starts to be quite cool, because we are starting having cross-research papers as well. So here, this is how Sergio starts his pipeline. So he has an MRI of vessels. He has also the functional response in the vessels. So has, not the functional response, have the flow in the vessels for the flow MRI. And what he does, he uses Xantao's approach to segment and reconstruct the model of the hemodynamic model, then calibrate it with 4D flow, and predicts some kind of flow that then he validates using ASL. Yeah? ASL, for those of you that don't know, it's a perfusion imaging, gives you something about the amount of ml that you have per unit of tissue per minute. Yeah? So validates with that, but also we'll receive some functional information that Gerling with his methods is going to process to give a more accurate ball now. And with our collaboration with the Linköping Institute University, in Sweden, we develop these models of the neurovascular coupling for different type of neurons that allow us to simulate ball from the hemodynamic, and then we optimize those with the ball that we observe. Yeah. So this ball is not only generated by hemodynamic, it's by a task given to the person, and with that we can infer which activation we have, which is the hemodynamic feedback, how the hemodynamics affect activity, and so on. So here you have an iterative approach in which you predict a ball, you compare with the original ball, and then you correct and try to get the best agreement that you can. So, and using this system, what Sergio wants to explore is how is the difference of the neurovascular coupling between patients that are in the control group and the patients that are undergoing dementia. And finally, but no less, is Stephen Kramer's PhD that is all about hypertension. We can talk about motivations of hypertension for half an hour because there are quite a lot. It's very bad to have hypertension in general. So let's submit that, and we can have then a one-to-one conversation if you guys want on that. But he's very intrigued by hypertension, and something that is quite interesting is hypertension, the knowledge that we have and how we treat it. Basically, if you are far from what is expected for you as being a human being in terms of central pressure, if you deviate off the normal, they say, okay, you have hypertension, we have to put it down. And how they put the pressure down, they try to put it as down as possible. So that is a little bit far from patient-specific and precision medicine and all the new contexts that we have. And ideally, what we want to know is first, why you have the hypertension. Is sustaining functionality? If it's sustaining functionality of the brain, we really want to get rid of that? So, one of the hypotheses that we are leveraging that can be related with dementia, not, for you guys, if you have someone with hypertension, as soon as they start taking certain medications, they go lightheaded, they lose cognitive acuity, so may that have an effect at all? Because the person seems to be hypoxic from the beginning of the treatment. What can be the long-term effects of that? So these questions are not known, and maybe we are completely going in the wrong direction, but it's very cool to research that, at least to know how the hypertension arises in a specific person. And second, what is the ideal target for that person if we understand why this person has hypertension? So, and by doing so, then we are not proposing here to develop any kind of drug treatment or whatsoever, but just to get a deeper understanding of what should be the ideal, and throw that problem then from our partners on the Faculty of Medicine, and then we'll figure out how they can elaborate a treatment for that. So as I said initially, this is a quite integrated system, and here we have the ANS model that is coupled with the heart, but also with the terminals in your brain, also with the carotid sinus and aortic arch. you have a heart model that receives a certain amount of blood flow from your veins, also receives some drivers from your parasympathetic and some sympathetic divisions that will change its behavior, will push blood into the arterial system. And then that amount of blood flow is going to be coupled with our nice vascular system. And again, we need to have some understanding of what is the peripheral resistance in the terminals, if that is going to be adapted by the ANS or not. And we have also some coupling at different parts of the system. So this is a very convoluted project, but we were able to do it because we start having people attacking all the different fronts. And that is where the integrative view start to happen. So we start having, for instance, CSF estimates of pressure from Alireza, so we can put the external pressure on our vessels. We start having the automatic methods that Chantal created to have this compiler to generate for each patient a patient-specific calibrated vascular tree that goes all in the heart axis. So we have all the information that we want for this kind of analysis. At the same time, with Cameron, we will start having a capillary level information that will allow us to have educated guess on the peripheral resistance, which is not easy to take patient-specific because it's a very hard work of trying to make some perturbation in the system to measure this. So this will give us a mechanistic educated guess. Also, if there is too much complexity in the geometry, we can use Harshild's models to account for energy dissipation and correct it in the right way to not underestimate it, as usually happens with low dimensionality models. And finally, we have also questions coming from our other more translational guys. Robin said, okay, we know that hypertension is developed early in people that were born preterm. So how these anatomical variations that these people usually have are involved in the development of hypertension? Which are the special traits that these people have? And we can treat them early in life to not have that? At the same time, we'll come the guys with more the neural activity stuff and say, okay, all this hypertension is super nice. Okay, it's not nice at all. But how that is changing our fMRI, what we should expect there is impairing activity. There may be another surrogate of how to detect hypertension when it's not beneficial. So, and this is the part that, as you see, I put all the people in my group in one slide. So that means that there is a lot of place of collaboration for these topics, and certainly one people cannot solve everything if you want to work in an ambitious project in this area. So that is our research side of things, that it's more translational and basic science, and we have also clinical studies happening at the same time. We have this study in Gisborne that is it's a region of New Zealand. It's not small at all. It's quite big. And here we have the opportunity of being close collaborators with the CEO of an MR center. And we want to run this Gisborne study on dementia that will take 100 patients through all our customized sequence on MRI to reconstruct these models and have a quite nice understanding of what are the biomarkers that we may derive from hemodynamics on these people. The second study is the newborn study executed by Robin, and that is getting this atlas for term and preterm babies. And she's getting more and more cases. I think that currently she has 20 subjects, and she plans to scan 10 more. That is a lot of patients for Robin, because imagine scanning a baby, and even more when it's newborn. So it's a very challenging task, and she's super good to do this. So cheers for her. And that's pretty much us. I hope that you can see why this integrated physiology modeling, we think that can be the next step on working forwards for diagnostic and treatment. Seldom are very few the problems that you can have in your body that is a collection of different systems intertalking that can be tackled with only one system. The good thing is are some of those, like plague and some very specific problems. But most of them needs of more than one system, and you need to start to getting your hands into fairly good physiology. And I motivate the question is, okay, more system, more parameters, and more chaos. Yeah, but it's quite cool to do that as an engineer point of view, but not always necessarily. So having more system is not that you need to have the same accuracy, the same level of description in your original systems. It's more about what are the parts of those systems that really matter. So yeah, I would really like to take this more to discussions and stop being only me talking. So thank you very much for listening, and let's see if you have any question.
Very good. Quite impressive. I mean, it's like you know about the brain a bit. Any questions from the audience? No? Yes? Well, I have a few. I mean, I can just start. You haven't mentioned anything about modeling the blood-brain barrier.
No.
Is this important for some of the applications?
Yes. So for sure, if you start working on drugs delivery, that will be the heart of the question for more cases. There are researchers on our institute working on that. That's why we didn't step in other people's toes. But yes, so it's important, the blood-brain barrier, and changed remarkably along diseases like dementia. Yeah, and if you have brain lesions as well, will change. But it's not where we are pointing our guns. But certainly, Sergio is doing something on that side. He needs to model it. He don't have any alternative at all. So
The neurovascular coupling.
Exactly, because in the neurovascular coupling you will have the blood-brain barrier here, yeah, and there will be things happening. So yes, we try to model as little as possible on that. It's not an easy area, and there are good people in the building working on that with new technologies based on diffusion imaging to try to characterize the level of the permeability of the BBB, yeah.
Mm-hmm. And also, I mean, here in the lab, we've been working with clinical collaborators a lot with fetal imaging and fetal applications. And I don't think you have shown use cases on this, that you have the term, the preterm, building this kind of vascular models. Are you thinking on, at some point, how to extend this to fetal vascular model and other kind of diseases where, I mean, these models could help?
Yeah, certainly something that can be done. We collaborate with Alice Clark, that she is super good in pregnancy and development. And we have the models that can, so if you go to the preterm, it's are variations of this one. As you go backwards, obviously you will have less amount of mature vasculature that you need to account for, but the junts are there, so that is the good thing, will not be that much of an issue. The problem is that the data that we have from the subject will be completely different, I imagine, because you need to start relying on MRI for everything in some way. For calibrating the model in a patient-specific way. And for sure, you are going only to see a few of the major vessels.
But what do you calibrate?
So here we use Doppler. For instance, we have from the babies, we have Doppler, we have pressure, and then we have a lot of ultrasound. So Robin scans all the arteries in systole and diastole and try.
There are ultrasound, I mean, Doppler, I mean, things in our models we're using Doppler in the fetal ones.
In the fetal one.
Yeah.
Okay, that would be very cool to discuss about and see what are the difference. Maybe everything is there already. Robin was working on this, and she was 100% of her time on that. Yeah. So we didn't have, I think, people to ask those questions really.
Okay. And you have mentioned at some point these AI-based surrogates of fluid in the vasculature.
Yeah.
So this kind of can you explain a bit more how it works, these kind of vascular slabs? How do you make this division? How local do they need to be? Is it important?
Yeah.
How local they
You want
are?
to make it as small as possible to not have very rich curvature and very complicated dynamics. That will be my advice to start with. So we try to make a bunch of them, but very small, because also GPU can parallelize a lot. So that is our mission. We are still defining which is the optimal size of the slab, which is the minimum amount of geometry variation we need to take in consideration for them to be general enough. So we are still at the stage that we are doing that. We have already, this is a ground truth that we have from TEPEN method, from Professor Blanco and Alonso, his postdoc. They are doing these lovely simulations with very fast, only minutes. So for the entire vasculature in the brain are a few minutes.
And it's a CFD, but surrogates of CFD with...
no, it's a full mechanistic CFD using a very interesting hybrid finite element that is spectral and P1. P1 in axial direction, spectral on the cross-section. And they can reproduce with a very nice performance the complete classic finite element method. And they have accelerated, but we know if we do our machine learning below that and then use the PEM to finalize the conversions, we are going to drop this one order of magnitude or maybe two more. So we are really after that to go in simulations of seconds for the entire brain vasculature. That is what we would like to hear, but let's see how it goes. But yes, it's in each slab. Then we do temporal decomposition because we assume that the flow is a transient flow, but we are going to assume that it's periodic. And because it's periodic, we can decompose in frequencies, and then you have NST state problems, depending how much is your temporal resolution you really want. And because of that, then you can approximate each one of the frequencies with an AI surrogate, as we proposed before. And these ones will account for different geometries. Sorry, different geometries and different boundary conditions. Yeah.
And you have kind of a lot of clinical use cases are kind of towards neurological diseases, or have you considered going the other way around, like to look more at cardiac diseases and the influence of the other systems in it? Arrhythmia, for instance? Or, yeah, I mean, whatever. I mean, cardiac failure or...
I think that all the infrastructure is there. So it's now through collaborators. And also Martin Nash group on cardio is going to throw us those questions at some point for sure. But we are super happy on explore those because all the infrastructure is the same. And as you said, it's only looking on the other direction.
And when you show some of these models, as you say, a lot of parameters, a lot of cows. I mean, in particular, these complete ones, I don't know, the hypertension. I mean, it's a lot of parameters on the ANS. How do you set them up? I mean, it just, well, sensitivity analysis, which data can you get just to personalize, I don't know, the ANS parameters?
The ANS, it's, by now, it's our biggest black box, and it's what we are putting most work-intensive research. I can tell you, so in the hemodynamic side, also you have a bunch of parameters. We have a very comprehensive set of MRI sequences that we run. So our 7 different MRI sequences, plus we have measurements of the sympathetic division of the ANS. We have also looking at urine samples to have an idea what's happening in parasympathetic. So, but we are still adding and adding at least more measurements until we have something super robust, and then obviously come the inverse process of trying to trim down what are the essentials and what is redundant information. The ANS is a great question. We have no idea yet. It's something to look at, and we are collaborating with people in physiology that they also do not know how to do that and are happy that we have models and they can try hypotheses. So I think that that is the way forward. We are in a complete unexplored territory, because we heavily think this is veins and nerves talking together. And obviously most of the people is not looking at veins for research. It's very hard to measure. There is not much money to do that as well. Who is going to put something on veins? It's very difficult. So I think that is where we have to look at, because it's where the ANS, it's modulating a lot, it's changing a lot the vascular tone of veins, and that will change your pressure completely. So I completely deviate from your question, I think.
No, but just discussion about the ANS and
Yeah.
That's interesting.
Yeah, so we think that maybe by looking at the veins, you are constraining more the system. You can infer a little bit better what is happening in the ANS. And what we are working a lot is Stephen is working a lot on heart models, but by our heart models that they need as input parasympathetic and sympathetic drivers. And because of that, you can start using all the information you know in the heart, like inotropy and chronotropy mainly. So this is the cardiac frequency and how heart is pushing blood flow. So you can make a guess using PV loops or even the cardiac output to know the inotropy. And with these two, these are have competing effects of sympathetic and parasympathetic. So if you know these two, at least you will know relatively how they are operating in relation with each other.
You are using a 3D model of the heart or just a LAMP model?
Currently, it's only a LAMP parameter model, and we are doing a little bit of phenomenological modeling. We are not completely happy with that, but it's what we can do with the data we have. We are having some interesting conversations with other people with bigger hearts to see if they can facilitate these monsters, because we are not going to develop a 3D model, with ALIA and these big models, what takes to implement that. But hopefully one of them just is keen enough to collaborate and we can put that.
It would be great to just, implant a device and see the effect somewhere else, right?
Yeah, that would be amazing. Certainly.
Very good. Any other questions? No? Just plenty of things. Really congratulations. Just super interesting and a lot of room for collaborations.
Yeah. Let's talk about that, yeah.